Hello, my name is Johar, and we are going to discuss about the enzymes. In the previous session, we have discussed about some portion of the enzymes. Usually, it happens in the normal situation. Some of the definitions, what are the categories in which it has been divided, and what are the factors that can affect the enzyme activity in a normal situation. However, in this session, we are just going to discuss about the inhibitors and poisons that can lead to various changes in the enzyme or the enzyme activity. So by the end of the session, we should be able to know, or we should be able to explain the different types of enzyme inhibitors and understand their mechanism of actions, how these inhibitors inhibit the enzyme activity, and we are going to discuss also about the regulation of enzyme activity, various kind of regulations. So the enzyme inhibition, by definition, an inhibitor is a compound that interacts with an enzyme to slow down the rate of an enzyme catalyzed reaction. As we have discussed in the last session, what is exactly enzyme do is basically to increase the rate of the reaction. So if anything that is stopping the enzyme or that is blocking the enzyme for doing its action, that is increasing the rate of reaction. So after binding with the enzyme or interacting with the enzyme, what exactly these inhibitors do? They slow down the rate of reaction, because increasing the rate of reaction is the main function of the enzyme. So anything that decreases the rate of reaction while interacting with the enzyme is an inhibitor. However, it has been divided, broadly classified into two major groups. One is the irreversible inhibitors, and the another one is the reversible inhibitors. What is the difference between these two is that even after we remove the inhibitors in case of irreversible, even if we remove the inhibitors from the system, enzyme does not recover or does not do its activity. Even if we remove the inhibitors. Why? Because the inhibitors form a covalent bonds with the enzymes, and they produce a structural abnormality in the enzymes that cannot be recovered. Therefore, irreversible inhibitor, in case of irreversible inhibitor, they used to deform or defect the enzymes that cannot be made working again, even if we remove these inhibitors. But however, in case of reversible inhibitors, they form non-covalent reversible interaction with the enzymes. So when we remove the inhibitors, enzyme can be reactivated, or they can their activity can recovers. Enzyme activity recovers when inhibitors is removed. Further, there are two major types of reversible inhibition. One is the competitive and the another one is the non-competitive. Let's have a look on the irreversible inhibitors. There are many examples of irreversible inhibitors. The cyanide poison, hydrogen cyanide. This is a powerful poison. There is an enzyme in the electron transport chain. We call it a cytochrome oxidase in the mitochondria. It inhibits the cytochrome oxidase. The cyanide, it is an inhibitor. It inhibits this enzyme, and there won't be any production of ATP, and there will be no electron transport chain. ETC will be prohibited or blocked. Another example is the fluoride. Fluoride is there is an enzyme enolase in the glycolytic cycle. We will discuss in the glycolysis about this enzyme enolase. This enolase gets inhibited by fluoride. So we used to put sodium fluoride in the vials. Sodium fluoride in the vials when we take the samples of the patients, when we do not want the cellular component of the samples to utilize the glucose, or when we want to accurately analyze the glucose level, then we inhibit the glycolysis by putting some fluoride ions in the sample. So this is also an irreversible inhibitor. Fluoride is an irreversible inhibitor of enolase. There are many uses of irreversible inhibitor. For example, aspirin, it is an inflammatory drug. It is a antipyretic, anti-inflammatory, and painkiller. There are three major functions. Along with that, it also being used as a for platelet aggregation, because it inhibits platelet aggregation also. So therefore, we used as a blood thinning agent in case of risk people or risk patient with cardiovascular problems or vascular events. Penicillin is an antibiotic. It inhibits the enzyme transcriptidase. However, the aspirin inhibits the cyclooxygenase enzyme. Another enzyme is the 5-fluorouracil. It is an anti-cancer. It inhibits the thymodylate synthase enzyme that is necessary for the generation of thymodylate. Now, reversible inhibition. Reversible inhibition may be competitive or may be non-competitive. First, we are going to discuss about the competitive inhibitors. Inhibitor binds reversibly into the active site. Active site means the substrate binding site. It is the same site where the substrate binds. And why? Because the structural similarity. If you look here, this is an inhibitor and this is a substrate. There is a lot of structural similarity between these two. So these portion approximately same in both the enzyme as well, the inhibitor as well as the substrate. Therefore, it competes. The inhibitor competes with the substrate for this active site, and sometimes it gets the opportunity to bind before the substrate used to bind. So when it binds with the enzyme, it takes some time to get relieved from the enzymes, because if it binds with the enzyme, it makes enzyme inhibitor complex, and it remains in this condition for some time, then again converted back into enzyme and inhibitor. Then the substrate might have an opportunity to bind with the substrate binding site and make the products. So in this kind of inhibition, there are certain important things to remember, that competitive inhibitors, first of all, it is reversible. Second, the mechanism of action is because of the structural similarity between inhibitors and substrate. Because of this similarity, inhibitors compete with the substrate. And the third thing is more important, is that what is the effect, what is the outcome of this inhibition, is that there is an increase in Km. What is meant by the Km, as we have learned in the last section, is that Km is the substrate concentration at which Vmax is half. I mean, the velocity of the reaction, or the speed of the reaction, or the rate of the reaction is half of its maximum at the substrate concentration at which the rate of the reaction is half of its maximum. So this Km is increased. What does it mean? It means that, as we learned in the last section, that if there is an increase in Km, it means there is an increase in the substrate concentration to achieve the half of the maximum velocity. It means that there is a less affinity of enzyme towards the substrate, because of there is a competition between substrate and inhibitors. Both can compete, so therefore both looks like the same, so enzyme sometimes gets confused and binds with the inhibitor also. So affinity of the enzyme gets decreased because of this interaction, because of similarity. The rate of reaction will be decreased, but if we increase the substrate concentration, then there will be more chance of only substrate that is getting chance to bind with the substrate binding site. Then we can still achieve the Vmax. So Vmax cannot be changed. As we remember from the last section, if we increase the concentration, then this situation is like this: substrate concentration and velocity. So here is the Vmax, this is the Km, and here is the maximum velocity. Okay, so if we increase the substrate concentration, then still we can. So this is without inhibitor, and with the inhibitor it will be like this. still we can make the maximum velocity at the same concentration. However, the Km is increased, like substrate concentration at which the half of the maximum velocity is increased. Okay. So this is about the competitive inhibition. Here is the diagrammatic presentation. Enzymes will bind with the substrate in the normal situation. Enzyme substrate complex will form, and then product will form, and enzyme will be released. Here, in case of enzyme substrate along with the inhibitor, because the structural similarity, it will have an opportunity to bind with the substrate binding site and make an enzyme inhibitor complex. However, this is also a reversible condition. It will again get back to this condition, and substrate will have an opportunity to bind with this active site, and then it will make a product. So the velocity of the reaction is decreased, but if we increase the substrate concentration, then the maximum velocity can still be achieved. Examples of competitive inhibitors are the statins. There are many statins, atorvastatin, lovastatin, pitavastatin, we take in to lower the blood cholesterol level, and it is a very important drug nowadays to for preventive measures for the vascular events, because if there is an increased concentration of cholesterol in the blood, there are more chances of vascular events. So therefore we used to decrease by using these statins. So because the similar structure with the HMG-CoA, they used to bind with the substrate binding site or the active site. Another example is the sulfur drugs. Yes, sulfonamide. Basically, these are antibiotics. Okay. Now the precursor will be converted into folic acid or into product. So when the enzyme act like this, the inhibitor acts, it will compete with this because of the structural similarity. If you see this, these are both of similar structure, this sulfur drug and the para-aminobenzoic acid. So it competes with this, and if in case of certain bacteria it needs this to form the folic acid, if we give this, the enzyme will this will compete with this for the enzyme, and there is a less concentration of folic acid will form, and bacteria will die because of lack of folic acid or decreased concentration of folic acid. So this is about the sulfur drug. How there is another kind of inhibitors, that is we know as non-competitive inhibitors. In the non-competitive inhibitors, it also binds reversibly to the enzyme, but at different site than the active site. Different site, we know the allosteric site. Like for example, inhibitors will bind here, this is the active site or the substrate binding site. The inhibitors might bind here or maybe here. For example, here it is binding here in this case. So inhibitor will bind here, and what happens? It makes some conformational change in the substrate binding site, so they have less affinity for the substrate now. So in this case, affinity is not decreased, but in this case affinity is not exactly decreased, but since there is less chances of binding with the substrate, with the enzyme, less chances of making the products as well. So Vmax will be reduced. It means that even if we increase the substrate concentration, it will not change the it will never achieve the Vmax. But in case of Km, that is the substrate concentration at which the half of the maximum velocity is achieved, it is usually not affected. It remains the same. So these are the summarized form of competitive and non-competitive inhibition. As we say that in the case of competitive inhibition, the substrate and inhibitors binds for the same active site, same site. However, in case of non-competitive inhibition, substrate binding site and inhibitor binding sites are different. There is isosteric site and this is allosteric site. So the binding site for competitive is substrate binding site. In case of non-competitive, it is allosteric binding site. allosteric site. And in case of competitive inhibition, the KM is increased. However, there is no change in the non-competitive inhibition. In case of Vmax, the KM, in case of competitive, there is no change. After addition of the substrate, we can achieve the Vmax, but in case of a non-competitive, it is decreased even if we increase the concentration of the substrate. So now the next topic is the regulation of enzyme activity. How the enzyme being regulated? There has to be some mechanism. Like for example, if we left the enzyme unchecked, it will convert all the substrate into the products. So we do not want this. We need products in a controlled concentration only. So for this, there are certain regulations. Sometimes we need reaction at a faster rate. Sometimes we need reaction at a very slower rate. So how our body used to regulate these things? There are many ways. Like for example, in a metabolic pathway, A is converted to E by sequential reaction. A is converted to B first, then C, then D, and then ultimately to E. So ultimately A will be converted to E. Now the E is the final product. This product, if it is made in high quantity, then it will affect the first reaction. This is known as a rate limiting step. Conversion of A to B. This is the first reaction. Rate limiting step or regulatory enzyme that is catalyzing this step. So this kind of inhibition is known as feedback inhibition, in which the final product will inhibit the first reaction or the rate limiting step or key regulatory steps. Any of the key regulatory steps is inhibited by the final product. This kind of inhibition is known as feedback inhibition. Now there are many kind of regulation. First is the rapid regulation, another one is the slow. In the rapid regulation, it can be allosteric or in the covalent modification. Like for example, allosteric, there may be positive effectors. It means that let's suppose this is an enzyme and this is the binding site and this is the allosteric site. So allosteric regulators binds with the allosteric site, and they either increase the activity of enzyme or maybe decrease. So positive effectors basically increase the activity of enzyme, and negative effectors basically decrease the activity of the enzyme. In the covalent modification, there will be addition of the phosphate group, that is known as phosphorylation, or maybe phosphate group is already being added and it has been removed by the enzyme, that is dephosphorylation. So either the phosphorylation activate the enzyme or deactivate or slow down the enzyme, or either the dephosphorylation activate or deactivate. It depends on the situation. So there are proteolytic cleavage converting. There are other type of activation. Third is the proteolytic cleavage. Like enzyme, there are certain enzyme produced in the form of proenzyme. Proenzyme, we call it zymogen. Like for example, pepsinogen. It will be converted into pepsin first, then only it can act. In case of pepsinogen, it cannot act. So it is a proenzyme or it is a zymogen. So there are three rapid mechanism. One is the allosteric, binding of any compound, positive or negative effectors on the other side than the binding side, and it might increase or decrease the rate of reaction. Covalent modification, there will be phosphorylation. There are many acetylation, methylation, phosphorylation, dephosphorylation. There are many types of covalent modification, and these modifications might increase or decrease the activity of the enzymes. And then there are another, that is proteolytic cleavage. Certain enzymes are produced in the proenzyme form, and then the name of these, we call it zymogens, and then it will be cleaved down or break down into some non-active compound and an active enzyme. This active enzyme will act on the substrate, and there is a slow or control enzyme quantity. This is a control activity, and this is to quantity. There is a slow method or slow regulatory activities. One is the induction and another is the reparation. In the case of induction, increased quantity of enzyme is being synthesized. In case of reparation, there is a decrease. If any enzyme is already being synthesized, the synthesis will be decreased or stopped. So this is known as reparation, induction and reparation. So rapid regulation, in case of allosteric, as we have discussed already, allosteric site, some allosteric regulators binds with the allosteric site, and after binding, there may be some, like for example here the allosteric inhibition is there. After binding the inhibitor at the allosteric site or the regulator at the allosteric site, there is some change in the substrate binding site, and substrate is unable to bind with the enzyme, therefore the enzyme activity will decrease. However, in case of activation, when the activator binds with the allosteric site, there is the substrate binding site will open and accommodate the substrate, and the product will be formed. So there are certain effectors that in case of allosteric effectors, it might be a substrate itself. Sometimes the substrate binds to its allosteric site and modulate the activity of the enzyme. In that case, it is known as homotropic effectors. And if something other than the substrate binding to the allosteric site and changing the activity of the enzyme, it is known as heterotropic. So homotropic and heterotropic. Homotropic is basically the substrate itself binding to the allosteric site also and modulating the activity of the enzyme, either decreasing or increasing. And in case of heterotropic, it is the anything other than the substrate that is binding with the allosteric site. But this kind of binding is not covalent, non-covalent. So it is not permanent actually. Okay. Now the feedback inhibition. Such kind of allosteric regulation takes place in the feedback inhibition when the final product used to bind with the enzyme of the first reaction and modulate the activity. Usually it inhibits. It makes certain changes so that the enzyme has no affinity for A, so no B will be formed. So this reaction will be stopped. Thus, such kind of regulation is known as feedback inhibition. Now, the covalent modification. In the covalent modification, like for example, this enzyme, it is a protein in nature, and then there is a hydroxyl group of serine or maybe threonine or tyrosine group. It is modified with a phosphate group. That is known as phosphorylation, addition of a phosphate group. Now this phosphorylation will either activate or deactivate the enzyme activity. Okay. Now the dephosphorylation will be done by the phosphatase enzyme, and phosphorylation is done by the kinase enzyme. So there are two different enzymes that either activating or deactivating the enzyme. So in case of this glycogen phosphorylase B, it is activated in phosphorylated form by the kinase activity, and it gets dephosphorylated when there is a phosphatase cleave out its phosphate group. So that's how the covalent modifications or regulation of enzyme activity happens. Now the proteolytic cleavage, as we had discussed, there is a spelling mistake here. It is N, it is not M. Limited cleavage of a small peptide by the protease from proenzyme to give as active enzyme. There are certain enzymes like trypsinogen and pepsinogen are synthesized. These are proenzymes. They cannot do anything, or they cannot work in any reaction. However, if it has been cleaved out some portion, like for example enzyme is this much big, and it will be cleaved out into this much and this much, 2%. This portion is useless, and this is an active enzyme. So this is pepsin. It can catalyze the reaction, however this pepsinogen cannot. So this is by the cleavage reaction. Cleavage happens here, the breaking down of one large molecule into two smaller. One is become active enzyme, and the another one is basically non-functional. But when it is attached with this, it will become all non-functional, like here. Slow regulation of the enzyme is basically induction and reparation, as we had discussed earlier also. Induction is the increased enzyme synthesis. Majority of the enzymes are proteins, so enzyme synthesis means translation of the enzyme, and reparation means decrease. Alteration enzyme level by induction on reparation of protein synthesis takes hours or days. It takes long time, so this is slow regulation. Induction and reparation result in change of total enzyme quantity. Enzyme quantity will be changed. However, in case of fast, there is no change in the enzyme quantity, only the activity will be changed. So elevated levels of insulin as a result of high blood glucose level lead to increased synthesis of key enzyme involved in glucose metabolism. As we know that insulin regulates glucose metabolism, so if anyone has a high glucose level, we used to give high insulin, or sometimes insulin gets released from the beta cells of pancreas, and it's used to induce many enzymes. It means that many enzymes related to the glucose metabolism will be synthesized. So that is all about this session. Thank you very much. Have a nice